Assalamualaikum warahmatullahi wabarakatuh to Doctor. Our group will be presenting about the upper respiratory tract microbiota of healthy adults is affected by Streptococcus pneumoniae carriage, smoking habits, and contact with children. So in this study, it discussed the importance of the upper respiratory tract microbiota to our health, and the study examines the nasopharyngeal and oropharyngeal microbiota of healthy adults aged from 25 to 50 years old, focusing and highlighting the effects of the factors such as the Streptococcus pneumoniae carriage, smoking habits, and contact with children, and how these factors affect the composition and interaction in the microbiota. And by comparing the microbiota of pneumococcal carriers and non-carriers and how demographic and lifestyle factors shape these microbiome communities, it improves our understanding of the factors influencing the microbiota and their implication to our health. Next, we're going to proceed to the purpose of the research. The first purpose is to compare the microbiota of pneumococcal carriers versus non-carriers in both the nasopharynx and oropharynx microbiota, and to understand how individual characteristics such as age, sex, smoking habits, and contact with children shape this microbiota. Next is the hypothesis. First, the composition of the nasopharyngeal and oropharyngeal microbiota differs significantly between individuals who are carriers of the bacteria and those who are not. Next, smoking negatively affects the diversity of the upper respiratory tract microbiota by increasing the number of potentially pathogenic bacteria. And lastly, adults in regular contact with children have a microbiota enriched in pathobionts commonly found in children. And next, we're going to proceed to the method of the experiments. The first step is the net study designs and the participant recruitment, which is healthy adults aged from 25 to 50 years old. And then we obtain the samples from the participants and store in the STGG medium. STGG stands for skim milk, tryptone, glucose, and glycerin. And then after the sample is collected and stored in the medium, it is then stored in the freezer with negative 80 degrees Celsius to preserve its microbial DNA. And then we do the DNA extractions followed by cysteine srRNA amplification, which involve PCRs in the V4 regions. After we obtain the applicants, we bring the applicants to the sequencing, which involve the next generation sequencing to obtain the data for the analysis.
Figure 1 shows the total bacterial load of nasopharynx and oropharyngeal sample. The oropharyngeal sample showed significantly higher bacterial load compared to nasopharynx in respective of the carrier state. Secondly, pneumococcal carrier has higher bacterial load in the nasopharynx than non-carrier. And lastly, the bacterial load difference underscore the distinct microbial ecosystem dynamic is in these two sites. Basically, this figure shows there are different bacteria species and total bacterial load different at these two sites. The second figure is volcano plot representing ASV that show differential abundance in nasopharynx. The findings are pneumococcal carrier has an increased abundance of Streptococcus, Haemophilus influenzae, and Fusobacterium nucleatum. Secondly, smokers show enrichment in Bacillus and Burkholderia, reflecting possible environmental contamination from smoking. And lastly, adults in contact with children exhibited higher level of Streptococcus and Marosella catarrhalis with decreased Staphylococcus aureus. 3 points of discussion for this article, namely the strength, limitation, and recommendation. The first strength is that this article provides a detailed comparison of nasopharynx and oropharyngeal microbiota, highlighting the effect of Streptococcus pneumoniae, carrier smoking, and child contact. The second strength is that the article employs a robust study plan, including longitudinal sampling, cysteine sRNA sequencing, and advanced statistical model, ensuring reliable and high-quality data. The limitations are this finding may lack generalizability due to the study focus on a specific geographic population, which is in this case Lisbon, Portugal. The second limitation is the relatively small sample size, especially among subgroups like pneumococcal carrier that limit the detection of final microbiota difference. The recommendations are to broaden the future research to include a diverse population across different regions age group, and health condition. And the next recommendation is to incorporate functional analysis like metatranscriptomic or metabolic to explore the functional role of microbiota in the respiratory health.
For the conclusions, this study reveals that Streptococcus pneumoniae presence in the nasopharynx shift the microbiota compositions, and factors such as age, sex, contact with children, and smoking habits shape the microbiota composition by reducing the diversity and increase the pathogenic bacteria in the microbiota. Therefore, we agree with the conclusion of the author because it is well supported with their findings which follow current understanding in the medical field. And the author also addressed the potential of additional unidentified factors which influence the microbiome composition in the conclusions. So that is all from both of us. Thank you very much.